Okay, so happy Monday. We're on week 11, I think. And also happy November. I really do appreciate that you're joining me tonight. We're approaching the last block, if you wish, of this course. This course can be divided into 4 sections, and we're starting today the last of these 4 sections that we'll be having a look of. And this section is going to be all about now that we have talked about the basic chemistry behind life, and we have looked at some of the basic molecules, basic structures that life is made of. We have looked at cells and some processes within cells, such as photosynthesis and cellular respiration. We're going to take all that, and we're still going to maintain that, but we're now going to kind of redirect our angle, and we'll be looking at cells, how they duplicate, so how cells divide, and a little bit of what sort of information drives that, what sort of consequences it has, and so on. Before I jump into any of the material from this week, and I'm going to be honest, I have a fair bit that we can go through, but we're going to go through only as much as it makes sense. So I have the material that's crucial for us to go through. And then I have, in addition to that, material that's going to be good to know if we get to that. So before I jump into that, I want to give a moment for any questions. So you can think at the back of your mind if you have any questions, and I'll take a moment for that in just a second. But before I do, I also want to remind, and I haven't looked who's present here today. We'll do the attendance in just a moment. But just in case we have someone present here who this might apply, I'm going to share this with you. It is important, and I'm not expecting that most of you would fall under this category, but I want to be, without calling anyone out, I'm just wanting to make a general announcement. It is important that you complete the assignments also weekly. If you only take the exams, you probably will struggle to get enough points for it to be worth it to get a good final overall grade. So it really is worth doing those weekly assignments. If you've been doing your homework and you've been doing your labs and then you've been doing your exams, you're doing the exact right thing. And I think that applies to pretty much all of the names that I'm seeing here. But I just want to be fair and make sure that I'm reminding, although I do feel that at this point of the semester, I hope that most would have figured it out. If you keep missing a lot of the work, it will be hard, if not impossible, especially if you're missing both homework, labs, and in addition to that, you're missing the exams. It will be hard, if not impossible, to pass the course. So I just wanted to put it out there to the universe, and not to be starting on a grumpy side, but I want to make sure that everyone has a chance to succeed in this class. And part of that succeeding in this class is that you know exactly what you're supposed to do. And I hope that everyone does. And what I see the names here, it seems very good. You're all doing a good job, but there's always one or two who just might sometimes get a little tired as the semester goes on or so on. So I just wanted to put it out there. While I'm pulling up the slides for today's class, I want to take this time for any of you. You can ask a question just by unmuting yourself and shouting out. You can type the question to the chat, or you can raise your hand. Any of those methods, and I'll make sure that we have a chance to answer those. Any questions about anything we've covered? Any questions about this week, about technical aspects, anything like that? In the past, when we were teaching face-to-face in a class, one of the things that we were always reminded was that make sure you take the time with the students to be quiet and give them the chance to ask those questions. And I feel that the time that it takes to ask a question has grown longer now that we're virtually. There's all these technical things, unmuting yourself, letting the microphone start, and so on. So if you didn't get a chance to ask a question, don't feel that you missed it. You can just give me a shout out at any part of today's session, or I'll always stay like I do behind at the end of the class as well. But I did not see any questions coming through just yet. What I would like to do is that we're going to jump to this week's material, and we'll look at two very key processes in cellular division. We'll talk about mitosis, and we'll talk about meiosis. And I'm assuming that you probably have heard a little bit about those in the past, in previous classes you've taken, or high school classes you've taken, or so on. And just to be safe, because sometimes things happen and I don't notice, you should be seeing now a shared screen of mine where there's a heading for the chapters 10 and 11. If you cannot see that, this is a really good time to give me a shout out. I'm going to stop sharing just for a second because I was supposed to do something. I have automated transitions for the slide when I did that for a recording, and I'm just wanting to make sure that they don't keep jumping around as we're going through this presentation. So hopefully that won't happen again. And going back to what I was saying, again, you should be seeing a screen of mine shared with the heading for this week's chapter. So before we dive into and talk about mitosis and meiosis in particular, let's take a step back and think just in general the concept of cells and cells dividing and all that. All organisms that are made of multiple cells, and that really covers most forms of life that we think. Most animals, most plants are made of multiple cells. They all start with just a single cell. And during the first round of division, they move from this, oh, I didn't mean to move ahead. Let's take a pen again. They move from this single cell during the first round of division into two cells. That single cell splits into two. After the 3 rounds of cell division, or second round of cell division, we end up with 4 cells. And after 3 rounds of cell division, we have 8 cells in total. By the time we get to about day 3 after fertilization, or cell division process in general, in humans, we are now roughly at the 4th round of divisions, and we have about 16 cells present. I know I haven't taken the attendance yet, and we'll take the attendance in a moment. So this is not yet something that everyone has to answer, but does anyone have any idea how many cells, if by day 3 we had 16 cells, how many cells would we have in a newborn human or human baby once they are born? How many cells might we be thinking that there are in a newborn? If you have any idea, you can just shout out or you can type it to the chat. It's not a trick question, and I will not feel bad if you don't know. We're talking about 2 trillion cells. So I'm just having to count my zeros here that I get them right. But that's a huge number of cells, considering that everything started here with just a single cell. Well, does anyone have any ideas how many cell divisions would it take? How many rounds of cells dividing for us to get to this 2 trillion? What's surprising to me is always that it's only about 14 rounds before we get to this high of a number. And there is a mathematical way to calculate this. And if anyone's interested, I think I share it in the recording for this week's videos, lecture videos, or I'm happy to stay behind. But at this point, just take it at the face value. It's not really that many rounds of cell divisions that we need to get all these 2 trillion cells that we need for the newborn. If we know that we have 2 trillion cells for the newborn, then I guess that the next logical question to kind of think would be that how many cells do we have for an adult? And of course, this depends on the body size of an adult and so on. But we're talking about anywhere between 15 and 17 trillion cells. So really, although the size of the baby grows as it grows into the adulthood, the amount of cells in relation to the original amount of cells when you're newborn doesn't increase that drastically. But what to me about this, at least to me about this story is always fascinating, is that remember all this, whether we're talking about 2 trillion or 15 or 17 trillion cells, all of that started with just one single cell. So in that first single cell that we had, there was instructions for everything that we see on the cells at subsequent stages of life. So that's quite a big and kind of an amazing concept to get your head around, at least to me. And I think I've shared with you in the past that I spent a bulk of my honors years in the university looking at the embryonic development. And that's really why I'm probably driven to consider these big questions, that how do we all start from one single cell, end up being so complex organisms, but also all of the cells at large that we have are at the right places. They're doing right things that they're supposed to do. Just by mere chance, one would expect that there would be more issues and more errors and more things that go wrong. But it turns out that that's not the case. We're actually remarkably successful in producing the final multicellular organism made of so many different cells with so very few errors in that process. So kind of to get us thinking about this, how amazing this cell division really is, I wanted to take a moment to talk about that. And I hope that if nothing else, I've conveyed my fascination for this process and hopefully kind of justified why these two processes are so important for us to try to understand at least the basics, because that's really what's driving everything that you will become, you have become, and you will be in a biological sense of the genetic material stored into the cells. I have a couple of other slides. When I was preparing today, I got really carried away, and I think I have a lot more slides than we have time to go, but what I'm showing here is simply this process of the first cell dividing into two, and then dividing further and further in subsequent cell division cycles. And the model organism that we used here was sea urchin. So really a simple animal made of multiple cells, but yet it's a perfect model to show everything that happens, whether we're talking about complex multicellular organisms such as humans or a very simple multicellular organisms. The basic concepts are the same. What I wanted to take as a chance for this to take the attendance is that let's take a easy question. And as you answered this question in the chat, that will serve as your attendance for today. So I'm asking that you to complete this statement. Multicellular organisms start as how many cells? Is it a single cell, pair of cells, so 2 cells? Is it a mitotic cell? Is it epithelial cell? What could it be? And if you were kind of following along the lines, you probably picked up that the message I wanted to share here was that we all start as a single cell. So if you had a chance to answer to that, no matter what your answer was, you are in for this class. Your attendance has been taken. There will be multiple chances. That's how I designed today's class, that there will be multiple chances to take part. And as always, more active you are, more I will then award at the end extra credit for having been involved in the class. It's important to take part even if you don't always get everything right. Grade A students don't always have every answer right, but they're actively involved. So thank you for everyone who answered to this question and got the attendance through that. I do have another question that's a little bit tougher. I don't know if you had a chance to pick it up. How many cells would we find after 4 rounds of cell division? And we use this model organism here, the sea urchin, but it could really be any model organism that we're looking at. And I see some answers coming through, and I believe that the answer that I suggested was that we would have 16 cells. These questions that I kind of incorporate to the classes are often similar, if not the same, to what you would see at the homework. I'm going to be honest, we're spending a little bit more time probably at the beginning than at the last questions of the homework. So at least you have a good chance of make a really strong start. What I wanted to take as the second kind of a thematic concept that we should think about when we think of the cell division is that really the difference between mitosis and meiosis. So many of you have probably heard about this in the past, so this might be just a reminder. For some of you, you might have forgotten it, so this is a good chance to now get acquainted with this concept for the first time. But not all cells divide alike. There's 2 groups of cells, and how they divide differs. And how they divide differs because these cells have a very, very different goal. And we're going to start actually at the end of what we'll look at. So we'll start by talking about gametes. And gametes are our sex cells, so either the egg cell that the female produces or the sperm cell that the male produces. So the goal of producing these sex cells is that we pass the inheritance material, the genetic material that we want to pass for our offspring. And by 1 cell from female and 1 cell from male fusing together, we get a new individual that has all of the genetic material that it is going to be made of. Half of it donated by mother and half by the father. This process of producing these sex cells is called meiosis. And let's have a look of why that's so important that we have a special name for this process. So of course, a new individual has to have 100% of the DNA of an individual. However, both parents cannot contribute 100% of that DNA. because if one parent contributed 100% of its DNA and another one contributed 100% of its DNA, and these cells would fuse together, then we would get an offspring that has way too much genetic material. It would have twice the genetic material of that an individual needs. So the special adaptation, if you wish to think of it that way, that happens in the production of the sex cells is that we're going to have to halve the amount of the genetic material produced in this process of producing either an egg cell or a sperm cell. So that's going to be a special process. And this process, as I mentioned, is going to be called meiosis. And we'll have a look of the process of meiosis towards the end of this session that we have today. And the reason why I'm saving it to the end of this discussion is that it's a little bit more complex than the process that we'll start with. So that was the one type of cell division that happens. This really doesn't include that many cells, only the production of the egg cells and sperm cells. So majority of cell division is going to be this other type of cell division. This other type of cell division that we're going to be talking about is going to be mitosis. Mitosis serves purposes, for example, when you have a scratch or your skin breaks, you get cut. All of these damages that happen, they will be fixed. So all of the tissues, by large, when you damage them, have an ability to be repaired. And the repair of tissues is going to be done with the process of mitosis. Also, regeneration of tissues. So cells cannot live forever. Every cell has only a certain lifetime. Cells rarely survive from the day when you were first born to the day when you're going to be dead. Instead, these cells over time get replaced when the old cells die. So we're constantly kind of going through this process of regeneration of tissues and cells. In general, I guess that you could describe it as a maintenance of the human body. So these cells, of course, need to end up having 100% of the genetic material, both in the cell that gives rise to the daughter cells and to the daughter cells, so the resulting cells that we produce. So both of these are going to have 100% of the DNA that we have there within each cell. So unlike in the process of meiosis, where we needed to halve the amount of genetic material, here we're simply copying everything that we have in that cell that's giving the rise to that copy. It's going to have the resulting cells. We call these resulting cells daughter cells. They're going to have the same amount of DNA in them. So that's why we're going to start with the mitosis. It's going to cover most of the cells, most of the cell division that we see in the human body, and it's going to be a little bit more simpler process. And once we totally master the process of mitosis, we're then gonna add a little bit more complexity and have a look what oddities do we find when we look at the production of the sex cells. There is one more thing I want to talk about before we dive in to the discussion of the mitosis. And that's some basic terminology that I think is going to be important for us to be familiar with. Just to make sure that we have shared language that we'll be using, we use the same terms, and whenever I use a term, you know exactly what I'm talking about. So I'm expecting a lot of this to be revision. There's some new stuff as well. So let's have a look through some of the basic terminology. So far on this course, we have talked about the cells, the basic form of independently surviving form of life. And within the cells, we had the nucleus, which was where the DNA, the genetic material of the cell, was housed. Often this nucleus, when we stained cells, stained darker, so it was also visually easier to identify when you did microscopy. Well, what we're going to do, we're going to now zoom in to the material housed within the nucleus of the cell. So let's do that as the next thing. What we end up finding is that at one certain stage of the cell division, the DNA within the cell condenses and it takes this shape of a structure that we call a chromosome. You might have seen chromosomes as these either X-shaped or H-shaped structures. It depends on what source you have looked at as which one they describe them. But chromosomes are really just condensed DNA and few other materials that we find within there. Chromosomes are not there within the cell, visible at all times of the cell's life cycle. They only become visible, they only appear during a stage of the cell division known as metaphase. And if at this stage of our discussion you're not sure what metaphase means, that's completely fine. We'll talk about metaphase in just a moment. So let's have a look of the chromosome. I mentioned that chromosomes are made of stuff. Let's have a look of what chromosomes are made of. Well, chromosomes are made of chromatin fiber, which is this kind of a bluish fiber that I've drawn here on this schematic representation of what's inside the nucleus. So the chromatin fiber often is found wrapped around these kind of bundles of proteins that you can see. And these bundles of proteins that we end up seeing are known as nucleosomes. So chromatin fiber wraps around the nucleosomes. And let's have a look of the chromatin fiber a little bit more. What do we actually find within the chromatin fiber? Well, we will end up finding that chromatin fiber contains our DNA. So often when we talk about DNA, and I think we had a chance to talk about DNA right at the beginning of the semester when we talked about some of the basic macromolecules that we find in life. So often when I describe DNAs to students, I remind them that DNA structure looks like that of a stepladder that has been twisted around itself. So it was this double helix structure. So double because we have these two sides that were connected by bases. And you remember that bases had to match from each side. And these bases were what varied. And then we took this whole structure and twisted it around itself, as you can see here. So there's going to be two strings, so double, and helix referring to the fact that it's kind of wrapped around itself. Well, if we look at a section of the DNA, so not all of the DNA, but a particular section of the DNA. Let's say that we're looking at this part of the DNA that I've kind of highlighted here. If we have a section of the DNA that codes for a certain protein or certain trait that we see, we call that a gene. So genes are sections of DNA that code for something that we can use, something useful. Often these are proteins or there are certain traits, certain characteristics that we find. And what I'm going to share next is kind of a reminder of what we discussed when we discussed about the DNA. So we talked about the nucleic acids and how the nucleic acids, DNA in particular, were involved with storing the genetic information. And then we had, of course, the RNA, which was the workforce that made something useful out of the information stored within the DNA. So it produced proteins. And when we looked at the DNA, we saw that DNA was made of nucleotides. So this is now as if we would be looking at a portion of this step ladder that we just saw. So we've taken a section of 1/2 of one step and the sides of this step ladder. So let's have a look of what those are made of. Those are made of nucleotides. So there's going to be multiple nucleotides that are connected to each other that forms the DNA structure. Nucleotides contained a sugar and a phosphate. So this sugar phosphate backbone is really which forms these sides of the step ladder, if you remember. And then these horizontal steps that you step on, I guess, these would be our bases. And you remember that our bases could vary. There were 4 different potential bases that we could have within the DNA. It could be either adenine, guanine, cytosine, or thymine. For RNA, it's the same with the exception that instead of thymine, we have now uracil. So really what we have thought here is just a reminder of something that we have looked at before. And what we're seeing is that the nucleotides are the building blocks of the DNA. And the phosphate and sugar were the sides, and then these bases were varied. And a right base on one side must match with a right base on the other side. There's only certain way these bases will match. So they form pairs where only certain bases attach to other bases. All that should have been revision. Earlier on there in this part, we saw some new terms potentially, and I think it would be a good time for us to kind of check how much did we catch from that. And what I have here is one of the questions from your homework quiz. And this question asks, which of the following is not true for a gene? So there's something that I've claimed here that's not true. Do we agree that a gene is a section of a DNA? Do we agree that genes normally, most of the time, code for a protein or a trait? Do we agree that genes are made of nucleosomes? So they have these sugar phosphate backbones and then bases. Do we agree that genes cover the entire length of the DNA? So is all of the DNA gene? And if you do want to have a go on that, you can, but I'm also going to show the answer. So we remember that, of course, gene was only a section of the DNA. Gene does not cover all of the DNA. There's actually a lot of what we call a junk DNA. They are coded with it, or junk DNA there within the DNA as well. So DNA material that does not code for anything useful for us. So you might ask that why would we have it then there? And I hope that by the end of the day, I will have given you an answer. Why would we have this junk DNA within the DNA then? Why is it useful to have that? What I wanted to focus a little bit more as we're kind of tackling this basic terminology was to take a moment to talk about chromosomes as well, because chromosomes are something that we're going to be looking at. We're going to be looking what happens to chromosomes as cells divide and so on. So again, it's going to be useful that we take a moment to make sure that we're totally comfortable with the language, with the terms that we use to describe different parts of the chromosomes. So you remember chromosomes were these X or H-shaped structures that condensed during the cell division, during the phase of metaphase. And let's have a look at what they're made of. So a single chromosome is made of 2 chromatids. So we have one chromatid here and another chromatid here. So in a single chromosome, we have two chromatids. Another thing that we want to notice from the chromosome is that these two chromatids are held together by a centromere. So centromere is this part, often central, not always equal distance, but often fairly central, connecting these two chromatid sticks or chromatids together. So we should be comfortable now with the terms chromosome, chromatid, and centromere. If you are totally lost, let me know and I'll try to re-explain. But this is something that I think is kind of a general thing that often still gets covered in the high school. What we're going to do, we're going to take this information that's a little general and we're going to add more to this information. And what I want to add is that I want to start talking about locus of a chromosome. And I just had to look there what's coming up so I don't get too far. So let's talk about locus of a chromosome a little bit. So every chromosome has a lot of material packed into it. And every chromatid contains a lot of this material. And what we find that traits are often stored or coded within the chromosomes at certain locations of the chromatids. So let's say we look at this location of a chromatid, and then we look at the same location on another chromatid. And what we end up finding, if it's these two chromatids are from the same chromosome, is that these two locations, so these two locuses, contain the genetic information for a same trait. So we see the same trait being coded twice in a single chromosome. So this location where this trait is coded in is known as locus. What I want to talk now is to take this concept of a locus and add a little bit more substance to it. Not both chromatids or both locations at these different chromatids necessarily code for the same outcome of that trait. Sometimes it might be that both locuses give us instructions to make the exact same thing. But sometimes what happens is that one of the chromatids, one of the locuses on the chromatids gives us instruction of doing one thing, and another chromatid on the same chromosome in the same locus gives us instruction to do a different thing. So these, what it codes for this particular trait in this particular locus are known as alleles. So sometimes we have the same alleles on both locuses, sometimes the alleles differ. So this might be a concept that you're like wondering now, okay, give me an example. It might make a little more sense. And if you were to say that, I would agree with you. So let's have a look of one trait. And this is only one example of many traits that make us all different. We all are a collection of multiple traits, but for the fact of making this describing what happens in genetic sense of our characteristics and different alleles, we're going to be looking at only one trait to start with. And the trait that we're going to be looking at is an allele that codes for the hairline of an individual. So you can have an individual who has allele for this kind of a hairline that comes down a little bit here at the very front. This hairline is known as widow's peak. You also might have an individual who doesn't have this widow's peak. Their hairline is all nice and straight. So now you might ask that, okay, we have these two different variations of a similar trait. Which one is more common? Well, it turns out that widow's peak, so this little part coming to the front, is actually more commonly found in individuals. This is because the characteristic for the widow's peak, the allele for that, is dominant. So if we have a dominant allele, we end up seeing that characteristic. It takes priority over the other characteristic. So whatever is the dominant allele, that's the one that we end up seeing, whether we inherit only it from only one parent or whether if we inherit it from both parents. So you'll notice that when I'm talking about dominant alleles in our classes, I'm often symbolizing them with capital letters. So in this case, I'm using capital W for the widow's peak. So if it's a dominant characteristic, that's common practice when we talk about genetics, is that we give it a capital letter symbol. Then we have sometimes characteristics, like I mentioned, this having a straight hairline, which are not as commonly seen in the population, but they're an option. These would be recessive alleles. And you'll notice that again here I've coded this allele for straight hairline, which is recessive, with the letter W, but this time I've used lowercase W for it because it's not as common. So whenever you have a dominant allele, even if you have just one of those, that's what we will end up seeing in the individual. So this is a good example of same locus on two chromatids of a same chromosome, and how the alleles of these same locuses can be different. You remember you had a dominant allele, which was the one that we see more commonly. That's always seen if it gets inherited even from only one parent. And then we have recessive allele, which is seen only if we inherit it from both parents. So to see straight hairline, you would need to inherit recessive allele from both parents. So let's talk about this a little bit further. So we can come up with 3 different combinations of alleles that were inherited from parents. And let's have a look at this first example where we had a widow's peak hairline. So in widow's peak hairline, you needed to have at least one dominant allele. So it could be that you only inherited this dominant allele from one of your parents, and the other parent gave you a recessive allele. You would still end up seeing a widow's peak. The other option is that you inherited alleles for this dominant characteristic from both parents, one from mother and one from father. In this case, both of these would be dominant, so they would be symbolized as capital letters. In the case where you inherited it only from one parent, it would be capital letter and the other one would be lowercase letter. So that's how you can get a widow's peak hairline. You have these 2 combinations of alleles, either 2 capital letters or 1 capital letter and 1 lowercase letter. So either 2 dominant alleles or just 1 dominant allele. Let's have a look of getting a straight hairline. The only way you can inherit a straight hairline is if you inherit a recessive allele from both of your parents. So both mother and father must donate to this new individual a recessive allele for a straight hairline. And that is the only way we will see a straight hairline in an individual, if both of the alleles for that are recessive. So now that we've talked about this, we see these three different sets, three different combinations of potential alleles that an individual might be left with, or might get at birth, might get at the point of fertilization from their parents. So rather than talking of a capital letter, lowercase letter, you remember that we talked about recessive and dominant alleles. Recessive was the lowercase, dominant was the uppercase letter. Well, what we're going to do now, we're going to look at these three combinations that we had possible, and we'll notice that the two first ones contain a matching pair on each of these alleles that we had. So we either had two dominant or two recessive alleles. So both of these are alike. If both alleles for a certain characteristic are alike, we call them homozygous. So homozygous, you can think of it two of the same. So homozygous, we have two same ones, one from each parent, but they're the same. So capital letter, or lowercase letter, lowercase letter. The only time that we can have a heterozygous pair of alleles is if we inherited one from one parent and another from another parent, and these were different from each other. So heterozygous alleles mean that we have different ones. So they're not the matching, these two alleles. So I hope that this makes sense at this point, and I'm kind of a little bit careful of what examples I pick, but some of my students in the past have shared that they found it useful when they think of different kinds of couples that we might have in this world. Most of the couples in our culture, in our society, are heteros. So we have male and a female. They're different in terms of their biological sex. You can have also homosexual couples where you have two of the same sex together. So if that helps you to think of that, good. If it doesn't, we're not taking this into a sociology class. We're not going to go any further down that road. So just be sure that you can separate the concept of homozygous and heterozygous alleles, and that you can separate between dominant and recessive alleles. If you're able to do all that, you're doing really, really, really well. And that brings us to a nice checkpoint before we're going to take a quick breather. So let's check how many of you got a chance to think of, and it's going to be numeric answer. A single chromosome consists of how many chromatids and how many centromeres? How many chromatids do you think that we have in a single chromosome, and how many centromeres do you think that we have in a single chromosome? Well, if you followed along, this should be easy. And I'm seeing good answers. So we have two chromatids. Remember, our chromatids were those sticks, if you wish, that we had in a same chromosome. And the centromere was that part that connected these two sticks together. So we're going to have two chromatids, one centromere in a single chromosome. I hope that that makes sense. I hope that that kind of is taking us back to where this all discussion started, and that makes sense. So let's have a look at the next statement. Do we think that it's true, or do we think it's false? A chromosome always contains the same allele for a particular trait on both of its chromatids. Will we always have the same alleles on both chromatids? And good job, I see answers coming through. And of course, we just saw that we could have different traits coded for same locus. So we could have different alleles. So you remember the dominant and recessive, you remember the homozygous and heterozygous that we talked about. So this statement would be false. Well, here's another one that's in your actual homework quiz. Widow's peak, is this statement true that widow's peak is a dominant characteristic? So offspring will always have a widow's peak if he or she inherits the allele for this trait from at least one of its parents. And the key word here is the dominant characteristic. An excellent job there. So that is true. If we have even just one dominant characteristic, we're always going to see that as the outcome of that individual. So great job. The next one is a chance for us to practice that language that we learned, and there might be more than just one correct answer. So which of the following are examples of homozygous alleles? Remember, homozygous meant two of alike. And if you answered both A and B, you were right. So as long as we have the same on both alleles, it is going to be homozygous. So what we see here for the C and D is going to be heterozygous. And very rarely would you see us writing, when we talk about genetics, a lowercase and an uppercase combination of alleles. Typically, we end up writing it as a capital followed by a lowercase if we write a heterozygous. So we don't specify which allele is from mother, which is from father at this point of our discussion. So if you tackled all of these, you did incredibly well, and you definitely deserve a breather. So what we're going to do, we're going to take just a minute or so to pause. We have now discussed the big concepts of the cellular respiration. And having discussed those big concepts, allow us then to move on to tackle the concepts of mitosis and meiosis. Before I jump into those, I want to take a moment in case anyone has any questions about anything that we have covered so far, so that there's a chance to tackle those. I'm going to be honest, I know our chat has been very busy, so if there was a question, I probably have missed it. I am scrolling through it, but if you did have a question, feel free to just shout out or rewrite it in the chat so that you get that answered. Well, at this point, I'm not really seeing questions coming in, so I'm going to carry on because we do have a limited amount of time together tonight, and I don't want to keep you over time. What I'm going to do, I'm going to jump in to the next section of this session together, and I think I'm going to spend just a little bit of time to talk about the cell division in general, and then, like I promised, we'll tackle the mitosis and meiosis. So at this point, you should be seeing my screen shared again, the entire slide, and this slide is labeled as mitosis. Although before we get to mitosis, there's one more little aspect of the story that I want to discuss. If you're not seeing the screen, definitely give me a shout out. So what we're seeing here is that we're going to move on, and we're going to first focus on the mitosis. So you remember, mitosis was what applied to most of the cells. So we're talking about processes such as repairing or maintaining or regenerating tissues. And these cells always have 100% of the DNA to start and to end. So the original cell that divides into two has 100% of the DNA. The offspring of that cell, if you wish, the resulting daughter cells, so the two cells that result from this cell dividing into two, both are going to have 100% of this DNA. So that's what we're going to be looking at first. But let's take a moment before we talk about that to get a little more terminology that's allowing us to talk about this process in a greater detail. So whenever I talk about cell cycle, or we talk about cell cycle in biology, we're talking about all of the events that take place in that single cell's entire lifetime. So like I shared with you at the very beginning of this session, pretty much all of the cells in our body get replaced throughout our lifetime several times. So cells don't live as long as we as organisms live. So cells get replaced as they get older, as they get damaged. And this constant process of repairing cells is what really allows us to avoid a lot of cancers, a lot of damage that occurs to cells, to tissues, that we can always repair it with a fresh new one that isn't damaged. So when we talk about the cell cycle, one way to look at it is to really think of it as a cycle. So we start with a cell, and when cell comes to the end of its life cycle, it might divide into two to give rise for two new cells. There is also an option that when a cell comes to the end of its life cycle, it just dies away. Because if every cell divided into two, we would end up doubling the amount of cells in our body with each cycle of the cells dividing. So not all cells always end up dividing into two. But we're going to be considering this lifetime of a cell, so this cell cycle, as this journey that this cell has, all of the different stages that it takes. And we're going to talk about three different stages in particular. So the first stage, and this stage is the stage that the cell spends a majority of its lifetime. This stage is called as G1 interphase. So during the G1 interphase, the cell grows and its DNA gets replicated. So the cell grows in size and cell organelles increase in number in preparation of that cell eventually dividing into two. So this is really the growth and DNA doubling stage of the DNA doubling is going to follow after that. So during S interphase, what we see is that now the DNA gets replicated. So we're making two copies of the DNA. So earlier on G1 stage, we just physically grew in size, and also the organelles, the cell organelles, were increasing in number, so that when the cell divides in two, we have enough organelles for both resulting cells. And it wasn't until the S stage when the DNA got doubled. During the G2 stage, which represents the shortest period of these three that we have discussed so far, the cell really keeps growing a little bit more in size, and it gets ready to divide. So now cell has grown in size. It has double the cell organelles, and it has double the DNA. So it's really ready to go this stage of dividing into two. And this division part is what we're especially interested in our discussion of when we talk about mitosis. And mitosis itself can be divided into this real division, if you wish. This real division can be divided into two parts. It can be divided into the stages of mitosis proper, where the DNA and nucleus get divided into two. And cytokinesis, where the cytoplasm gets divided into two. But together, the mitosis proper and cytokinesis are just called as mitosis. And this is the stage that we're especially interested in our discussion today. However, before I jump into mitosis, I do want to share with you that one of the reasons why we don't have so many cancers, one of the reasons why cell division works so well and we don't have those errors, and why it's so successful, is because we have these checkpoints. So checkpoints are stages of the cell cycle where the process cannot move ahead unless the needs and demands of that checkpoint have been met. And there's going to be three checkpoints that we're going to be talking about. The first one is going to be G1 checkpoint. During G1 checkpoint, this is the primary or the most important checkpoint, if you wish. Cell is committed to dividing after passing through this checkpoint, and during this checkpoint we can check for the DNA. Is it how it should be? If there are errors in the DNA, we can fix it. And if we cannot fix the errors in the DNA that we have at G1's checkpoint, the other option is to stop, to arrest the cell cycle, and none of these later stages would take place if the cell does not pass this first checkpoint, G1 checkpoint. The G2 checkpoint, which we're going to talk about next, this really verifies that the DNA has been replicated. And again, if we have errors, we can fix the DNA here as well. Of course, if the fixing of the DNA is not successful, we simply stop the progress of the cell cycle onwards. And the final checkpoint, which is going to be our mitotic checkpoint, to move on from here, all of the chromosomes of that cell must be attached to a spindle. And you might wonder, what is this spindle that he's talking about? And you're going to see that in just a second. So for the cell to continue further into the stage of mitosis, all of the chromosomes must be attached to a spindle. So let's have a look of all that. But before we do, another quick quiz question that I have put aside here. Which of the following statements about cell cycle would not be correct? Does a cell cycle contain all of the events that take place during cell's lifetime? Does the cell cycle include G1, S, and G2 interfaces? Can cell cycle start to reverse if a checkpoint is not passed successfully? And if cell cycle is completed successfully, the cell ends up dividing into two daughter cells. So which of these would not be correct? And if you answered C, you are right. Cell cycle does not start to go backwards. It progresses only on one direction. And if we have errors in some of these checkpoints, we just pause there. We don't move ahead, but we do not go backwards. We can try to fix things at that point, but we do not go backwards. So now, after all this preparation, we're finally ready to talk about mitosis. So we remember mitosis was all about cell division that relates to the maintenance of the tissues, maintenance of the cells, repairing tissues, repairing cells, regenerating those, keeping our body in a good shape. In all other parts of the body, with the exception of the production of the sex cells. You will find a pretty nice diagram that's very similar to what I'm going to show to you next. What I'm going to show to you next is one that I've drawn, but there is a similar diagram that's provided by your textbook manufacturer, publisher, and you can access that on board. I've made a link there for you for that. So if you find that be useful, you can refer to that. Do we have any questions? I think I just heard myself. I don't hear any questions, so I'm just going to jump into the mitosis in the interest of time. We're going to look at the different stages of this process called mitosis. And there's going to be some names of these stages that might initially seem a little strange. And really the best way to learn them is by simply memorization. There's not that many stages to learn, really. And if you take the time to learn the stage and the name of it and what happens in that stage, you're going to be golden. So what we're starting with is a cell that we see here. And this cell has, of course, the cell membrane. And inside the cell, we can also see the nucleus. What's worthwhile to notice here is that we have a clear nuclear wall that separates the nucleus from the rest of the cell. Now, when we move to the first stage of mitosis, this stage is going to be called prophase. During prophase, the nuclear envelope that earlier on surrounded the nucleus disappears. We're now seeing our chromosomes floating around free in the cytoplasm. And we see two more structures. We see centrosomes. And you might remember that I talked about centrosomes back in the day when we went through the cell organelles. But I talked about them only as very briefly. And at that time, I said that, oh, you'll hear loads more about centrosomes once we get to talk about cell division. Well, today is the day when you'll get to hear more about the centrosomes. So they play an important role in the process of cell division. So centrosomes appear on both ends of the cell, both poles that are going to be giving rise to two separate cells. And from the centrosomes, we can see these fiber-like structures coming out. These are known as our spindle. And spindle is going to be an important structure as we move further into mitosis, in a sense that they are responsible for controlling the movement of our chromosomes. Remember, at this stage during prophase, the chromosomes are still floating around free within the cytoplasm of the cell. So they're not really organized, they're not really attached to anything. They have appeared because the nuclear envelope of the nucleus disappeared and the DNA condensed to take the shape of a chromosome. But the chromosomes are floating free, just these two new structures that we're seeing. We're seeing centrosomes and we're seeing spindles coming out from those. Well, let's move on to the next stage, which is going to be called metaphase. During metaphase of this mitosis, what happens is that now these chromosomes are attached to our spindle, and they align nicely on this line that goes halfway through the cell. We call this line an equatorial plane. So if you think of the geography and the Earth, we have an equator around the Earth, bang on in the middle. It forms the middle line around the Earth, separating the northern and southern hemisphere. So that term and that concept is related to that. So we have an equatorial plane in the cells that are about to go through mitosis as well. And this is thanks to those spindle arranging, helping to arrange those chromosomes to get organized. What happens next is that we move on to anaphase. And during anaphase, the single chromosome is divided into two. You remember each chromosome was made of two chromatids. So now each chromosome is pulled apart so that one chromatid goes to one end of the cell and another chromatid goes to another end of the cell. So they are going to be identical in sense of the locus that we have, but of course the alleles and so on could differ. We'll have a look of these differing alleles in the production of the sex cells, but for our purposes, we're just going to think that these chromatids are exactly the same at the process of mitosis. The other thing, other than the chromosomes being divided into two, with the chromatids being pulled to the different sides, other thing that we're noticing is that the cell, and this is still the same cell, but now it's starting to give on the outer surface, on the cell membrane level, first signs of dividing into two. So this cleavage starts to form. So this cleavage is only visible in animal cells. You remember animal cells had as kind of a, if you wish, a more moldable, softer, more moving cell membrane, whereas plant cells were much more rigid. So rather than cleavage forming in the plant cells, what we see in the plant cells is that they start to grow a new wall, but only part of the wall is visible here. But in animal cells, we can actually see this indentation where the cell will be snipped into two. And now we get to the last stage of the mitosis, which is going to be called teleophase. In teleophase, we now have two cells that cleavage has continued and eventually snipped the cell into two. You remember the chromatids were pulled to the different ends of the cell, and now the nucleus and nuclear envelope reappear. So we really have in every sense two new cells that all originated from one initial cells. We often call these cells as the daughter cells. So if you want to summarize what happens in mitosis and the names of the stages and what happens in each of the stages, we started with the cell, then prophase, where the nucleus disappears, the centrosomes and the spindle appear, and the chromosomes were floating free in the cytoplasm. During metaphase, the chromosomes lined up on the equatorial plane. During anaphase, those chromosomes were split into two chromatids, one chromatid to each future pole of the cell, and the cleavage started to form. And finally, we get to teleophase, where we now have two resulting daughter cells. The nucleus has reappeared on both. And if we compare these resulting cells, they are exactly identical to each other. They're also identical to this initial cell that we had, that we started with. And this would be a good chance to kind of test what we learned. Who remembers, is it true that during prophase, the nucleus of a cell is visible? Remember, prophase was the first stage. And one of the big things that we saw during the prophase was, of course, that the nucleus disappeared. The nuclear envelope disappeared. So now that we have talked about the process of mitosis, I'm going to come back to what we just talked about, the cell cycle. The cell cycle, there's two different signals that can control how far the cell cycle proceeds. And we're going to look at three of these. The first one of these is going to be external stimulation. So the cell can be stimulated to keep dividing, and it can be stimulated by certain hormones, certain other chemicals. These are often known as growth factors. So these promote for the cell to go ahead and work towards dividing into two. Unlike stimulation, we can also have inhibition. Inhibition refers to the forces that would signal to cells that now it's time to stop from dividing any further. And a good example of inhibition that we sometimes see is when we put cells into a petri dish. So petri dish is this glass container that's open from the top. It's very large. It has normally some sort of a growth media where these growth cells are going to grow nicely. But as these cells grow nicely and multiply and take over the cell, eventually they will end up touching the walls of this petri dish. And this physical contact with the walls of the petri dish promote inhibition of the cell cycle. So now we're sending a message that it might not be in the best interest of these cells to keep dividing. Let's stop the division process because there's no more space within that petri dish. Another way that we can have to control the cell cycle is a limit to the division numbers, how many times we are dividing the DNA or the cells. So you can think of it as a comparison of taking a photocopy of a document. So here we first see the original document. It's nice and clean. When we take a photocopy of a document, there's little errors that start to build up. And more times we take a photocopy of that document, more errors we are left with. So our cells and our DNA have a very clever way to get around that. And this is where this junk DNA steps in, like I said. So what we're seeing here, I've taken the DNA that we saw earlier on, and I've just drawn it out as a one single stick. And these orange areas would be, in this example, genes that code for something particular, something useful. Everything white on this DNA would be this junk DNA that doesn't code for anything useful. You'll notice that there's especially lots of junk DNA at the very ends of the DNA. And every time that we make a copy of a DNA, it just so happens that we are not able to copy the few very last bits of the DNA. So few very last bases are not going to be copied. So every time the length of the DNA, and especially this junk DNA at the end, is lost a little bit. And as we keep making copies and copies and copies, we have less and less of that junk DNA there. But we never want to get so far that we fail to copy some of the important DNA that codes for a particular gene. So for that reason, we have a limit to how many times a cell can divide, because the DNA, with each time that it divides, we lose some of that. So like I said, you can consider it that we end up building errors as we keep copying the cell, the DNA again and again and again. And this having a limit to how many times the cell can divide, which refers to also how many times the DNA in there gets copied, this helps us to avoid that we never get so many divisions that the actual genes that are important for us don't get copied correctly. And also it helps us to get away from having too many errors building up, so we don't get genes that would be faulty or do weird things that they shouldn't do. So those were 3 examples of how we control the cell division. And there is actually kind of relating to that what I said about the limits of the division of the copies. There is a small section at the certain part of the DNA. Once we reach to that point, which is highlighted on these diagrams as this dark green section, we make no more copies of that particular DNA. That cell is no longer going to divide more. So kind of a checkpoint question here. We have discussed about the cells stopping the division when they reach the edges of the Petri dish. Which of the following cell cycle regulatory signals would this be an example of? And if you were with me, I know that we're kind of approaching the end of our section, but if you were with me, you probably picked it up that that was an example of inhibition. Also thinking of the photocopy concept, I'm asking, does taking a photocopy of the DNA improve the quality of the resulting print? And of course it does not. Every copy copies also all the errors that we have ever had. So that's why there's a limit. I know that we have reached a time where really we are supposed to wrap up for today, but I do want to go through the process of meiosis. So if you have to go, I completely understand it. If you're able to stay, I'm probably going to try to go through this rather fast, but I do want to get through everything. So I do want to talk a little bit about the process of meiosis and how that differs from everything that we have just learned. So let's have a look of that. So we remember that earlier on we were talking about mitosis. Most cells, the replication of those was done through process of mitosis, where the DNA amount remained as 100%. Well, when we are producing egg or sperm cells, so we're producing our sex cells, we only wanted half of the DNA material to each of the resulting sex cell, because you're going to have 2 sex cells, the sperm and the egg cell, that are going to fuse together. So 50% from each is going to lead to us having 100% in that final resulting offspring. So I've done there all sorts of nice animations. Another way how we often describe about it when we talk about genetics is that we talk about n, and a lowercase n symbols 50% of the genetic material. So a full individual would have 100% of the genetic material, and that's often symbolized as 2n. So if you see 2n, you know that we're talking about 100% of the genetic material. Single n referred to 50%. So there's 2 special concepts as it relates to the process of meiosis. The first concept that I want to talk about is the concept of synapsis. In synapsis, chromosomes, 2 chromosomes come next to each other, so they pair up. So 2 chromosomes pairing up is known as synapsis. And crossing over instead refers to the process where the parts of these chromosomes that have now become synaptic get exchanged. So little bits of these chromosomes get exchanged as a process continues and the crossing over takes place. If you think what would be the purpose of the synapsis and crossing over, you might realize that this is introducing us more genetic variation. So we're getting more different kinds of random combinations of potential DNA that we pass to the offspring. So we want to really, in the production of a new offspring, We want to hit that genetic jackpot. We want to come up with a new individual that has the best possible genetic makeup. And only way to hit the genetic jackpot is to try to get as many different combinations as we can and hope that one of those combinations is that jackpot. So we're doing everything we can to introduce variation to the genetic material. So what I'm going to do, I'm going to talk you through the process of meiosis. And what you'll notice is that meiosis is very similar to what we saw in the mitosis. The big difference here is that we talk about prophase 1 and prophase 2. We also talk about metaphase 1, metaphase 2, anaphase 1, anaphase 2, and telophase 1, telophase 2. So we basically go through the cell division process, but we go through it twice. So we're going to start at the very beginning of the story, which is going to be prophase 1. Here, again, as we saw earlier on, the nuclear envelope will disappear, centrosomes will appear, and spindles will appear. The big thing to notice here is now that the synapses and crossing over take place. So the chromosomes come next to each other, so they form chromosome pairs, and parts of their contents get exchanged. And this was, as I said, all about mixing the genetic material, introducing as much variation as possible to this genetic material. We move on to the metaphase 2, and now we have chromosome pairs. So notice it's going to be pairs of chromosomes that line up to this equatorial plane. I've only drawn 4 chromosomes, so 2 chromosome pairs on this diagram, just to keep it a little bit clearer and simpler. Now, when we move to anaphase 1, the chromosome pairs separate, they're pulled to the different poles of the cells, and you'll notice the chromosomes will be different at different ends. The cleavage, which we saw earlier on, that will start to form. Now we reach the stage of telophase 1, which is actually only a halfway point of the process of meiosis. The nucleus and nuclear envelope have reappeared, and cell has divided into 2 resulting daughter cells. However, remember now that the chromosomes are different on these 2 cells. You saw how the chromosome pairs, they were different, the ones that ended up to different poles of the cell. So we're going to have already variation on these resulting daughter cells. They're not going to be alike each other, and they're not going to be like their original parent cell. Well, now we go through a second round of divisions. Important thing to notice here is that during this second round of divisions, we are now not multiplying before that any of the genetic material that resulted onto these 2 daughter cells. Since we are not doing any multiplication, any duplication of the genetic material, we are going to end up with only half of the genetic material on these resulting daughter cells. So on this first round, we did have the full 100% of the genetic material at the end, but at the end of the second round, we're only going to have half of the genetic material on the 4 resulting daughter cells. This process goes through the same stages we've seen. Again, the prophase 1, prophase 2, prophase 2 is followed by metaphase 2, anaphase 2, and telophase 2. So now at the end, we actually have 4 different daughter cells. And remember, each of these daughter cells is going to have a unique combination of genetic material. So they're all different from each other, and they're all different from the parent cell. So if we summarize the process of meiosis, you just see that there's two cycles without duplication of the genetic material. So we go to the half of the genetic material, and this important step of having synapses and crossing over taking place, so we can introduce as much variation as possible to these resulting daughter cells. So if we are comparing the mitosis and meiosis, we remember that mitosis was about all other cells than sperm and egg, whereas meiosis was only about production of the sperm and egg. Mitosis contained one cycle, whereas meiosis two cycles of cell divisions. Mitosis resulted in two daughter cells, whereas meiosis in four daughter cells. In mitosis, the genetic amount was 100% for the original cells and resulting cells, whereas at the end of meiosis, the amount of genetic material is only going to be half at the end of it. And this is where the terms haploid, referring to the half of the genetic material, and diploid, referring to all of the genetic material, come in. Also, we've already touched upon how at the result of the mitosis is two identical cells, and not only being identical to each other, they're identical to the parent cell. Whereas in the result of meiosis, we had four each unique combination of genetic material on these cells, and these are going to be unique also in comparison to their parent cell. So that really covers all of the big concepts that I wanted to talk about today. I am going to throw out one concept that's kind of covered in a greater detail in your lecture videos, and this concept is going to be karyotype. Karyotype is a method how we can view all of the chromosomes of an individual cell. And this is a useful test. It's a very basic test, but it's a useful test for finding out a lot of information about potential errors, especially on babies that are early on their development after fertilization. So we're going to be looking in a karyotype the number of chromosomes, we're looking size of the chromosomes, we're looking if there's any abnormalities in those chromosomes. And of course, that allows us also to see the sex of the individual. So here we are seeing a karyotype of an individual. So we see all of the chromosomes that we have there, and we see that we have 23 pairs of chromosomes. The chromosome pair 23 that we can see here, that's going to be coding for the sex of an individual. And you remember if it was XY, that's going to be male. And if it's going to be XX, that's going to be female. So we can use the karyotype to tell the sex of an individual, but we can also use it to tell if there's any genetic abnormalities of a baby that the parents are expecting. And I think that I normally go into quite a bit of detail. Students are normally quite interested about these abnormalities, but that's not something that I would test you on. Depending on how we get along on our Wednesday session, I'm happy to provide you a little bit more information about some of the genetic abnormalities that we can pick up with karyotyping, but I would not expect you to be masters of that. I would not ask that in an exam. And that really completes everything that I had planned to cover for today. I know that we ran a little longer than I had anticipated. So if you have stayed all the way to the end, I would appreciate it if you drop your name to the chat. And that completes our session today. You are free to go. I will be providing also a recording of this. If you have any questions, you can stick around and I'll be here to help and answer those. I know it was a lot of information, but you'll find out that the rest of the semester is really going to be just using this information and building on it. So I appreciate you having joined me for tonight, and I hope you have a really good rest of the night, and I hope that your November has gotten to a great start. Have a good night. Thank you. Yes. Did you have any questions? Can I help with anything?
Yeah, I have one question. So
Sure.
when it comes to the symbol N, would it best describe mitosis? N?
Sorry, I totally missed the first part of the question.
So does the symbol N, just N, which is describing the resulting cells, the DNA content, would that describe mitosis?
You could use it on either one, really. And let me jump here to the slides. Sorry, I think I got so carried away with having so many slides. So you could use it also when you talk about mitosis. But remember, in mitosis, it would be always 2N because it's going to be 100%. In meiosis, we move from 2N to 1N because we're halving the genetic material. So I'm not trying to confuse you, so let me try to rephrase it. So the N refers to 50% of the genetic material. So what we see in meiosis is that we move, and I'm trying to take a pen out, sorry. In process of meiosis, we move from 2N, which was 100%, to simply 1N, which is 50%. But nothing prevents you from using this N symbol when we talk about mitosis. In mitosis, it would just always be the 2N. We go from 2N to 2N again. Does that answer, or did I just confuse it more?
No, it makes sense. So, just to make sure, a single sperm cell would be N, right?
Yes, that would be
Okay.
correct.
Okay.
And single egg cell would be N. So what we're producing in the process of meiosis was that we wanted to produce a sperm or an egg cell where we had only half of the genetic material. So that's why I can see where you're coming with the question. I think I should have highlighted that before. So a single egg, single sperm is always going to be only 50% of the genetic material.
Okay, that makes sense. Thank you so much.
I so appreciate that. Was an extremely good question, and me it shows that you're thinking all the right things. I think you're going already above of what I would expect you to think. So what you're thinking is putting you probably to the next course. So good job.
Thank you. Have a good night.
Thank you. Have a good night. I see Riley on the class. Did you have any questions? Is there anything I can help you with? I'm going to make a wild guess and just assume that you didn't have questions. If you do have questions, just jump in. I'm going to pause the recording at this point and just take notes so I get the attendance for today. But don't hesitate to speak up if you have any questions later on. I really appreciate everyone who joined me also through a recording for this session, and I hope that you have a very good rest of the night. Thank you. Bye.